Thank you very much. So welcome back, everybody. My name is Dr. Melanie Hamilton. I'm joining you from the Jane and Ron Graham Center for the Scholarship of Teaching and Learning at the University of Saskatchewan on Treaty 6 territory and home of the Métis. And I really just want to thank Sean for being invited, and it is such a joy for me to participate in anything to do with organ donation. Organ donation is very near and dear to my heart. I am a mom of a transplant child. I had a daughter who had a liver transplant in the middle of the pandemic in 2021. And it's hard to believe that we're coming on 4 years since her transplant, but I'm happy to say she is well and living her very best life post-transplant. But what makes my story more unique than most transplant families is I'm a nurse by discipline, and I actually worked in the operating room at the U of A from 1999 to 2000. And I worked on the donor team where we harvested the organs from our donors, but I also had the privilege of working and being a scrub nurse on some of the transplants that we did over the 2 years that I was at the University of Alberta. And the craziest part to my transplant journey, or mine and my daughter's transplant journey, is when I met the surgeon who was going to be doing my daughter's transplant. It happened to be Dr. James Shapiro, who I worked with in the operating room 22 years prior. And I will tell you that it made it so much easier for me to send my daughter to the OR because I knew what was going to happen in the 6 hours. She was undergoing her liver transplant. But more importantly, I knew she was under the care of Dr. Shapiro, and it just made me feel really calm and confident that she was in great hands. And that's kind of my full circle moment of how important organ donation is. But right now, it gives me great pleasure to introduce a very special guest speaker joining us all the way from Melbourne, Australia, and she is also joining me on Zoom this afternoon. Professor Karin Verspoor is the Executive Dean of the School of Computing Technologies at RMIT, University of Melbourne in Australia. She's a fellow of the Australian Academy of Technological Sciences and Engineering, a fellow of the Australia-Asian Institute of Digital Health, and a co-founder of the Australian Alliance for Artificial Intelligence in Healthcare. She was named a 2021 Brilliant Woman in Digital Health and selected as a finalist in the Women in AI Australia New Zealand Awards in 2022 for AI in Innovation. Karin is very passionate about using data and AI to improve health outcomes in people. Her work has specific emphasis on the use of natural language processing to transform unstructured data in biometrics, ranging from scientific literature into clinical texts in actionable information. Dr. Verspoor's presentation is entitled Opportunities for an AI-enabled scientific knowledge, exploration, analysis, and discovery. Please join me in welcoming Dr. Verspoor for her presentation this afternoon.
Hello, everybody. Thank you so much for having me. And yeah, this will be a little bit of a change of pace, I think. Before I start, I'd like to acknowledge the traditional custodians and the ancestors of the lands and waters where I'm joining you from today, which is the lands of the people of the Woiwurrung and Boonwurrung language groups of the Eastern Kulin Nation. And so I'd just like to respectfully acknowledge all of the traditional owners of the lands where you are as well. So yeah, I'm here to talk to you about something maybe a little different, which is the use of AI in the context of our day-to-day activities in science. And where I'd like to start is, of course, with the research literature, right? So all of our science, by definition, is grounded in knowledge, and that knowledge is communicated through scientific research publications. So we do our research, all of the things that you were talking about today, and of course we can communicate it in presentations like you've been doing, but the standard by which we contribute these things to the community is by writing it down and putting it out there in some form of publication. And you'll be aware that we have kind of hierarchy of evidence, and at the very top of it we have our systematic reviews, which tries to integrate and synthesize all of the research that has been done around particular clinical questions. And generally speaking, in the context of medical research, we focus on randomized controlled trials and systematic reviewing of those randomized controlled trials as one of the key places where we derive our medical evidence. And that means that pretty much every study revolves around a pretty structured question. So the structure of a randomized controlled trial can be boiled down to what we call the PICO framework: the population, intervention, the comparison or control group, and the outcome. And almost every study can be framed in terms of a statement like, to assess the effects of the intervention compared to the comparator for a condition or problem in a population in some context on outcomes. And obviously the population can be quite carefully defined, but most randomized controlled trials essentially boil down to this sort of statement. And if we keep that in mind, then we can think about how that translates into finding the evidence that helps us to understand that the interventions and their relationships on to outcomes in certain populations, and can bring us into the realm of looking for that evidence in the literature. And typically what we do is we use these structured query terms. So any of you who've searched in PubMed are probably familiar with the process of basically trying to put keywords in there and maybe as you can see in this example, putting in some spelling variants or, adding in extra terms that are going to give you some context, maybe using mesh headings, the medical subject headings that are associated with papers that are indexed in PubMed, and so on. And we use this process as a typical way that we drive the systematic review process. Now, one key problem is that the research literature is just huge. And, so this was a snapshot I took just a few days ago, but, PubMed now in 2025 has more than 38 million citations indexed in it. And the graph on the right tries to sort of catalog the growth of that literature over time. And it's just amazing how much research there is out there. So actually the process of doing a systematic review, even when you're looking for fairly constrained questions, is pretty daunting. So the talk today is really to walk you through some of the different ways that we might be able to leverage the tools of computation and AI to help us do a better job of this. And I won't say it's a solved problem, absolutely not. But there's a number of things that people like me have been working on to try to make the research literature a little bit easier to find and navigate. So let's start with trying to find things in the first place. So one of the key things that we all know about the literature is that it's a lot of words, right? And words can be ambiguous, and there are all different ways to express the same kind of concept, which makes it quite difficult to boil down what we're looking for into those structured query terms that we can use in PubMed. And so some of my work has been around trying to map the words into concepts or entities and events or standard identifiers so that we can help to organize the information that's in the literature a little bit more effectively. And just to give you an example, this is a sentence from a corpus that we built more than a decade ago, actually, called the Colorado Richly Annotated Full Text Corpus, where we manually went through and identified key terms in biomedical literature, in this case, molecular biology literature, relating particular genetic mutations to particular biological properties, and mapped those key terms into ontologies. And we use the sequence ontology, we use the gene ontology, we use the cell ontology, and we used entree gene identifiers. So we try to deal with the fact that there are all these different ways to express particular concepts and boil down the information to the concept. And then we tried to automate that process by learning from the human annotations and using the ontologies themselves to map into a more structured space. And this is important because terms like membrane budding have realizations in the literature in all sorts of forms. And some of our controlled vocabularies, like the gene ontology in this case, do capture synonyms, and they capture some of the variations in these terms, but they don't always map neatly to how they're actually used in text. And particularly, you might have a term in the gene ontology that gets realized as a whole sentence or as a verb instead of as a noun phrase. So membrane budding can map into vesiculate or be related to that. And vesiculation process is another way of referring to that. So some of the terms will map neatly into the ontology and some of them won't. And so we were trying to come up with ways to essentially recognize all of the variants you see on the right, so that we could map it into a controlled identifier. You can do the same thing with the Unified Medical Language System, which is a massive, let's call it an Uber ontology or an Uber vocabulary of basically any term you can imagine that's relevant in medicine. And we want to try to map the detailed complexity of language into these structured terms. Another example where we focus specifically on the PICO structure, the population intervention control and outcome structure, is work that was done by some colleagues of mine, where they went through the literature again and tried to identify all of these different types of information as expressed in the literature. And this provides us with a foundation for trying to automate these things. You can bring these things together. So during COVID, we did some work where we created what I call PICO concepts over the literature, where we try to identify those broad categories of population intervention and outcome, but then have more specific concepts from the Unified Medical Language System or from the medical subject headings vocabulary, and essentially transform a sentence like cumulative COVID-19 related hospitalization and death rates into a concept which says, okay, we have an outcome that's described in this phrase, and specifically we have two outcomes of interest: hospitalization and mortality. And if we do that across the entire literature, we can build a database which then allows us to search by concept rather than by words. Another example where we go actually beyond concepts and into relations can be found in the context of things like protein-protein interactions or chemicals treating diseases, mutations causing diseases. Essentially, we can capture core facts that are expressed as conclusions in scientific studies or as key findings in a study. And by transforming a sentence like cyclin E2 interacts with CDK2 in a functional kinase complex into a structured representation that says, okay, we have a protein-protein interaction. The first interactor is cyclin E2, the second interactant is CDK2. And by the way, that protein-protein interaction results in something that we can label with gene ontology identifier 9358, which corresponds to the concept of polyphosphate kinase complex. And if we do this across the literature, then we can start to do more structured. We have more structured access to the information in the literature. We can also incorporate knowledge. So all of these concepts and terms come from a controlled vocabulary or an ontology, where sometimes we have information like categories of things and higher level concepts. And so we can interrelate the concepts to each other by leveraging the knowledge that's captured in an ontology. Here's an example of some work that we did around trying to identify chemical-induced disease relations. And so again, we have, a title and an abstract with lots of words in it, but we can boil it down to those basic facts of, okay, we have a relationship between a particular chemical and a particular disease. And that just makes the information that's communicated in all of those words a lot easier to see, certainly from a computational perspective. And that makes it easier to search. Another context where we've done this is in the context of chemical patents. And in this case, we were actually trying to structure the entire reaction information, the chemical reaction information that was expressed in the text. And so that means boiling down, again, a complex sentence like the one at the top here into these very structured concepts and relationships. And in this case, we were mapping those chemical terms into categories of starting material or solvent or reagent, and then looking at the triggers for particular steps in the reaction. So once we have done this process of organizing the kind of entities and relationships, the concepts and relationships that are described in words in the text, words and sentences, then we can put it into a database, and it makes it a lot easier for us to search for example, papers that correspond to particular interventions of interest, in particular populations with particular outcomes. And, it's rather than you having to come up with all the different variations, all the different ways that these concepts and relations might be expressed, you can search in that structured space using the controlled vocabulary terms, and it just makes it a lot easier to find things. Of course, we're now in a world. So I've been doing this work for quite a long time, more than 20 years by now, and we're now in a world where we have generative LLMs. And so you might think, well, we don't need any of this fancy, old-fashioned natural language processing anymore because we have even fancier large language models that can do all of the work for us. And I guess there's still some questions around how exactly that will work and how well generative LLMs will do this job. So one of my undergraduate honor students actually did a project where we just tried to get the LLMs to do this job of information extraction, trying to identify genes and variants and diseases. Basically, we actually tried to do it in Spanish. So we participated in a challenge task last year where we try to get the LLMs to annotate this biomedical information over Spanish abstracts. The LLMs did an okay job finding things. I wouldn't say great, but this was what we were trying to do, was to transform the text into that kind of structured list of the entities that were expressed in there. And as I said, we did this in a cross-linguistic way. So we played with actually prompting it in English or prompting it in Spanish, and we tried different ways of doing the prompting. And we provided the instructions, or we didn't provide instructions. So we played with all these different ways. Just to keep the story short, we found that there were actually a huge number of hallucinated instances. And overall, the results, if you don't have to look too carefully, but overall, the results were sort of, around 0.6. Generally speaking, over here, we have it maxed out at 0.5. So not doing a great job of finding everything we want it to find, and then over generating things through hallucination. So I guess the story there is LLMs are not quite ready to do this job, but obviously they will improve with time. So I guess watch this space. So we also found that they mostly the hallucinations were in the forms of instances that were not that could not be mapped to every instance in the text. So basically the LLMs kept on telling us, yeah, this disease is there, this disease is there, this disease is there, even though it was only mentioned once in the text. And there were several instances of entities that were just not found in the text at all, which is strange because they have access to the text. We weren't trying to hide that information. Okay, so once we have all of this structured representation of the information that's being communicated in these scientific papers, how do we want to make it available? Well, in the examples I was giving earlier, we generally speaking knew exactly what we were looking for, right? We have that PICO structure. We know what kind of information, what populations, what interventions, what outcomes we're interested in looking for evidence about. But if you wind your head back a few years to the beginning of the COVID-19 pandemic, we had quite a different situation. Part of this problem then was that, just like with other science, we had massive accumulation of research that was coming out. But one key difference that we had, and so this is 2020 back here. So you can see that already in March of 2020, we were starting to get, I don't know, that's about 400, 500 papers every week coming out. So massive amounts of science being produced about COVID-19. And we realized that actually there was just too much information, and scientists, could no longer find anything. And I had the pleasure of speaking to a journalist at Science Magazine about this, and I kind of made the point that, well, we've been trying to provide tools for scientists to help find information. These aren't really new tools, but it's good that scientists are now discovering that they need text mining. So I was kind of happy. But one thing that was really clear at that time is that the questions that people had were not neatly PICO questions. They were not in these neat structured frames. These questions that the World Health Organization published were extremely open-ended, right? What has been published about medical care in COVID? What do we know about vaccines and therapeutics? We didn't know much in the beginning. What do we know about the COVID-19 risk factors? And so rather than having these really clearly structured questions, we had these open-ended questions, and the tools of Medline searching don't allow us to ask questions like this. And so we started thinking about how we could leverage the tools of information extraction and trying to organize the biological or the biomedical knowledge in a way that would allow us to move into supporting these kinds of more open-ended questions. And the normal mode of searching is sort of summarized here on this screen. You know exactly what you're looking for. You have a well-defined information need. You do that game of trying to get your keywords just right to find the things, and then you get back some results. You kind of look through the first 5 or 10 of them, and you think, okay, yeah, that looks interesting, and that looks interesting. Obviously, if you're doing a more systematic review, you would do this more systematically and review each one. But, in general, you're relying on the quality of the query at the outset in order to drive this whole process. And when your questions are more open-ended, you're going to get back way more than you can possibly screen in a reasonable amount of time. So what is beyond that kind of paradigm? Well, why not take advantage of these categories, these concepts, these relationships, and even summaries and synthesis that we can get out with our computational tools? And so by transforming the documents into those things, we can then imagine a different way of engaging with the literature. And we developed a prototype tool, which sadly we've been unable to maintain since COVID. But anyway, so you can't actually play with it. But the idea was to build on those concepts and relations in order to organize the information and then visualize that organization. So we use those PICO concepts I told you about earlier, ran it over all the COVID-19 articles we could find, and produce this kind of visual overview of, hey, we've got some population terms, we've got some intervention terms, we've got some outcome terms, and here's how they're connected. And you can actually, in the system, you could click on one of these links and it would pop up the paper that actually had that relationship in it. And so this was a different way of making the scientific findings accessible to people that didn't involve knowing exactly what you were looking for at the outset. The overview here actually surfaces the concepts that are in the literature rather than you saying, I want to look for this concept. Okay, so then we have the problem of trying to synthesize all of that literature, and in the process of systematic reviewing, we can also think about leveraging natural language processing, or AI, not just for that kind of searching stage or the exploration stage, but also for supporting the other aspects of systematic reviewing, including summarization and quality appraisal. So we've been doing some work along those lines, and I'll start with some very recent work, actually, by an epidemiologist who's actually an associate professor at the University of Queensland, and then came to me and said, I want to learn about NLP so that I can write my own tools to help me with systematic reviewing, which I thought was really special. Not many people who are already associate professors decide to come back and do another thesis. But anyway, Gary's been working on trying to build tools that help him to do his systematic reviewing. And so the problem that we've been starting with is title and abstract screening, and trying to leverage not LLMs per se, but a small language model that allows us to model the statistical patterns in the literature. And so what we've been doing is to try to use the specification of a systematic review, including the title, the objective, the selection criteria, and the review background that we have going into the systematic review, which is usually defined in a protocol. Use that protocol background to allow us to automatically screen the medical literature that's retrieved. So if we retrieve literature using the kind of normal keyword searching approach, we can use then the automated methods to help us decide whether one of those papers that we pulled up is actually relevant to the systematic review specification. And so we looked at Cochrane reviews, and we used that essentially as training data for developing a system that allowed us to deal with both the sort of the more obvious screening of excluding literature after just title and abstract screening, and then the slightly more subtle problem of full text screening, where an article looks relevant on the initial title and abstract screening, but then is identified to be not relevant after screening the full text. And so this work is really about using the representation of all of the words in a paper and comparing it to the representation of all of the words in the specification to allow us to decide, is this relevant or is this not? And we try different ways of doing that. And the upshot is that we can actually do quite a good job in terms of recall or sensitivity at a good level of specificity. And we were able to show that this holds across a wide range of review topic areas. And so we're continuing work along these lines, hopefully to further improve the performance of these tools. And who knows, maybe they'll be useful for a systematic review that you do one day. This is just a little bit more detail on how we do that. As I said, candidate paper compared to the systematic review specification. And these little gears here are trying to reflect how we're modeling these individually. And then this bit is about doing the comparison and making a prediction about whether to exclude the paper and at what level. Okay, so in terms of the summarization side of things or evidence synthesis side of things, some work by my student Yulia was really looking at how we could leverage natural language processing to help us do that summarization. And one key point that Yulia makes in her work is that you have to synthesize, not just summarize. And so if you have a collection of literature that has superficially related information like these statements here, you actually need to dig in a little bit more deeply in order to understand what the overall assessment is around, in this case, a particular PICO question. So we have our PICO question of allosterin. Does allosterin lower blood pressure? So is the outcome of lowering, and the blood pressure is the population in a sense. And if you just look at all these statements here in the middle, you'll see that actually there's quite a lot of variation in the findings of the papers here. Obviously, this is just an example. But what we see is that, we have to at least have the PICO question, right? We need to know what the clinical context is. We need the direction of that, which gives us a proposition. And then we need a modality. So may, does, doesn't, right? And that all these elements together form the claim that is described in the paper. And if there is no modality, so a statement like, thus it is important to establish if allosterin lowers blood pressure, semantically does not communicate any kind of claim. And so we need to be able to rule those things out. We can aggregate at the level of PICO and basically say, well, this study said this, and this study said that, and this study said that. That is certainly possible to do, although it can be quite tricky to do that in a concise way. But most of the systematic review contexts actually require aggregating sometimes quite conflicting things, because the population definitions are often a bit different, because the quality of the studies are different, because the findings and the outcomes are different. And so if you think about trying to summarize a bunch of apples and oranges and pears, you can just list them all out. Like I said, this paper found this, and this paper found that, and this paper found that, but that actually doesn't help us to draw any conclusions about it. You can take the majority vote, right? 3 out of 5 papers had this finding, and so, well, we'll say, okay, yep, that the majority says the finding is positive, and so we will conclude it's positive. But actually, in practice, what we want is something more subtle, right? Some say orange, while others say apple, and some say pears. And in fact, maybe the right way to synthesize it is to say, well, it's oranges, even though orange is not the majority finding, but perhaps it's the highest quality. And so thinking about how you get a machine to do all that reasoning around the quality of evidence, around the best way to synthesize the information, and even to detect the direction and the modality of a statement is quite complicated. What do you do when there's no evidence, or there's too much conflict, or all the evidence is weak? How do you aggregate weak evidence to strong? Can you even do that? All these are questions. I'm not saying we have answers, but it's important to recognize the complexity of this decision-making process. So another aspect of the systematic reviewing is the quality of evidence assessment, and that would play into how we approach the summarization and synthesis of the information. And this was tackled by a research fellow, Simon Schuster, who looked really at that problem of, okay, how do we assess the quality of evidence? Because we can't really do that synthesis without understanding the quality of particular individual studies. And so we need to have a strategy for determining the confidence of our estimate about the effect of an intervention on an outcome, while considering the trial design and the conduct of the research itself. And so we again used Cochrane reviews as our context, as the exemplars of the process of synthesizing evidence. And that's because Cochrane reviews use a framework called GRADE, which allows, which involves assessing the quality of evidence for each outcome that is reviewed across studies, and to come up with a rating of evidence, high, moderate, low, or very low, in the process. And so that framework looks at different aspects of the studies: risk of bias, inconsistency, indirectness of the study, imprecision, publication bias, and comes up with a grade. And so we developed a machine learning approach, which basically used the words in the text to help us make an assessment of those quality criteria and come up eventually with a grade score automatically. And this is still, imperfect. People are better at this than machines are, but we were able to show that, using the representation of the documents, that we were able to do at least some reasonable grade scoring. So rather than using that whole manual effort, we wanted to move towards an automated method of trying to do the prediction of the different aspects of the quality. We also tried using large language models because, of course, like I said, we have large language models now. Can we use them to help us address these kinds of problems? And so we've been using large language models and asking them to do the job of grading the quality of evidence in a particular study, and also to look at the possibility of having those more specific questions, like this one here: were participants aware of their assigned intervention during the trial, in order to get more fine-grained assessment of the quality of particular studies. And so we prompted LLMs with the signaling questions and with the text themselves, and try to get them to address the quality of different aspects of the study. And the bottom line is that even at a binary classification level, like doing risk of bias prediction of yes, there's bias, or no, there's not, the LLMs were doing pretty bad. So on par with very, very trivial baselines. And it didn't really matter what kind of strategy we used to do the prompting. We found that it just didn't work very well. Again, it's a moving space. We're limited to using large language models as they appear, but these kind of higher level analyses of the literature are still hard for LLMs to do. So we're making progress on this, and whether we continue to use LLMs or whether we revert back to these kind of more targeted ways of approaching and modeling these tasks remains to be seen. I think we still have some more work to do. Definitely the human brain and the ability to synthesize information has not been replaced by AI just yet. Okay, so now just moving on to the last topic around evidence discovery. Once we have all of the information in the literature captured in that structured form with concepts and relations, you might think of this as a knowledge graph. So trying to pull together those concepts and relations. Think of the nodes in this graph here as different concept types. So these might be genes, and red is diseases, and what have you, and the connections between them are shown as links. That's basically the process that we do when we transform the literature into more structured representation. Once we have that, we can start to look for patterns within that graph. And so we did some work, again, kind of in coming out of COVID, where we were trying to look for associations between drugs and look for drug combinations by doing a network analysis of effectively where things clustered in this graph by looking at the context of use. This was really exploratory, but it was trying to show that you can leverage the structure of these relationships to help you do some inference. One line of work is related to something that we call literature-based discovery, where we're essentially trying to fill in the gaps in that big knowledge graph. And this concept actually dates back to the 90s, where a man named Don Swanson proposed the idea of combining information from different parts of the literature together in order to generate a hypothesis about things that might go together but haven't been shown in the literature yet. You can think of this like your Netflix recommendations, right? If you have watched a movie which has certain characteristics and other people have watched other movies, if you like this one, other people who like this one, you might like this one, right? So Netflix does this, Amazon does this. Imagine doing this in the context of the literature. We're actually looking at doing that at scale now. Back in the 90s, they were doing it one by one. So the example that Swanson gave was, okay, there's a paper over here that shows that fish oil is connected to managing high blood viscosity platelet aggregation. And we know from another part of the literature that high blood viscosity platelet aggregation is connected to Raynaud's disease. And so Swanson put these ideas together and said, could fish oil perhaps be a treatment for Raynaud's disease? It's a hypothesis, and then it can be tested. And in that particular case, it was in fact shown to be true. And, that was done in a fairly targeted way, but now we can use the tools of big data, computation, and these knowledge graphs that we can build out of the literature to do this at scale. And so my student Gracie Poo has been working on exactly that. Okay, I want to talk a little bit more about the LLMs because, I said our LLMs to the rescue. I've given you a few examples where LLMs are not quite working as well as we would hope. And I want to also highlight some of the more challenging issues that we see in using LLMs in this way. And this is some work, again, that Gracie has been doing around intervention, extraction, that structuring of the information. And essentially, we've been working with some researchers at the University of Edinburgh who work in Alzheimer's disease and specifically in preclinical animal studies. And they had been using a regular expression or dictionary-based way of trying to find names of drugs and these sorts of things, and finding that it wasn't working very well. And so we experimented with trying to use ChatGPT, GPT-4, to help us do this process more effectively. And we essentially found that using ChatGPT in combination with the regular expressions allowed us to improve the performance, particularly in terms of recall and finding more things that were relevant. But on the other hand, the performance over here, none of these performances are anything to write home about. So, again, not really a well-solved problem. My student Tin was working on negation, and this was actually inspired by work that we were doing over clinical text, recognizing how important negation was. And Tim has been able to show that the language models effectively don't see negation. So what do I mean by that? So here, we have a multiple choice question, should be pretty easy, right? The normal respiratory rate for an adult male is one of these three answers. The correct answer is C. Well, Tin just played around with negating the question. So the normal respiratory rate for an adult male is not. And guess what? The LLM gave exactly the same answer. And in fact, this should be even easier because 3 of the 4 options are correct, and it didn't give any of them. So the baseline is sort of 75% accuracy if you're just randomly guessing, and the LLMs simply couldn't answer the questions properly. So, the cute title of the paper, language models are not naysayers, but the point of it was that the LLMs really struggle with negation. And I can explain if you're curious about why that is, but essentially it's because negation is not context specific. You can find it anywhere, and because it has a particular semantic effect, the LLMs just don't even, they're not working at that higher level of understanding. Another example, does a man owns a dog entail a man owns an animal? Yes, a man owns a dog entails a man owns an animal, since a dog is a type of animal. Okay. Does a man does not own a dog entail a man does not own an animal? Yes, a man does not own a dog entails a man does not own an animal. Well, maybe not. So yeah, again, LLM struggling with negation. Now this is improving, and Tin actually came up with some strategies for trying to get the LLMs to pay more attention to negation and understand that it did have semantic implications. So we will probably see this, it's again, an evolving space, but it's important to understand that these models are not necessarily behaving in the way that we want them to behave. And even more capable models with more targeted focus on negation are still have plenty of room for improvement. Another example of this, which has to do with numbers, extremely important in our clinical texts and in systematic reviews, right? Everything, populations are defined by numbers. This is work by my student Ramad, and he's been looking at basically how the LLMs can do numerical reasoning. And again, you don't have to look too hard at the numbers, but most of them are below, or around 0.5. That's not very good. And so the ability of these models to do inference over statements that involve numbers is quite weak. More numbers. And this is particularly important in the context of our biomedical texts, because so many of our definitions of our populations and even interventions relate to dosages and age information and vital signs and so on, which are all numbers. And if we can't reason effectively over those numbers, then we can't make sense of the data. So as an example, we have most patients in cohort 1 suffering a cardiac event. And in order to interpret this correctly, you have to be able to do things like aggregate. Okay, well, these are all cardiac events. We've got 1 here in each of these categories. That's 4. Well, 4 out of 93 is far less than half. And so this is actually a contradiction. The models are having trouble with those sorts of reasoning. Yeah, so the LLMs are experiencing a lot of difficulty, particularly when there's confounding information. Okay, so I will leave it there, but the basic upshot of all of this is that we can really leverage the power of natural language processing and AI to help us make sense of all of the information that is out there in this massive amount of literature that we have. It really can be helpful. Whether LLMs are the right solution, I think time will tell. But we are trying to constantly work on tools that can help us to navigate and make sense of all of the scientific information that's out there. So I'll say thank you, and I'm happy to take any questions.
Thank you so much, Dr. Verspoor, for sharing such an interesting topic. It is just fascinating to learn about artificial intelligence. We can now take questions from both online or in the room. And if you're asking questions in the room, there are microphones you can use, so those of us that are online can hear. And if you're online, you can type your question into the chat, and I can ask the question to Dr. Verspoor on your behalf.
Stunned silence. That's okay too. Give you all food for thought.
Well, thank you very much for this fantastic talk. My question comes from the perspective of being a researcher who is very interested in the use of AI in science. And my question is also, could be philosophical, around the ethics of using AI, especially in hypothesis testing. So how, as a user, as an end user, how can I not cross the thin line between ethically doing my research through writing my own manuscripts and using AI to hypothesis test and generate manuscripts. Because part of your talk was about like using AI and using algorithms to find material for systemic review writing. So I was thinking, if I used AI to do that to me, what's left for me to generate as an author?
No, that's a really good point. And I should be clear that, you know, we're definitely not trying to fully automate any of these processes, right? So each of the the kind of steps that I presented are intended to help you do a more effective job of doing the systematic review work that you're doing. And, you know, recognizing that there are limitations, like I said, to keyword searching, there are limitations to, you know, how efficiently you can review. Systematic reviewing, like the whole process, can take years to do properly. And, you know, in fact, I just reviewed a systematic review yesterday, and they had this statement in the paper that said, oh, you know, we executed the search on this date, and the next day another study came out. And I was like, well, just include it, right? You know, you need to have an ongoing updating. And the reason we don't do that is because the tools that we have require so much manual work still to do it. Now, you will never hear me say we need to replace the scientist with a machine. You know, as I hope I've made clear, there are still a lot of questions about how well these tools can do the job of understanding numbers, of understanding negation, of synthesizing information. And so what we're trying to do is to help these machines that you're going to be using anyway do a better job of supporting you, but not at all replacing you. You still have to come at it with your critical lens, and you have to calibrate what you know and what you've read with the output of any tool. I hope that makes sense.
It does. Actually, there's another question of, is disclosing using AI to generate like systematic reviews or manuscripts can undermine the effort of a researcher?
I don't think so. I mean, look, there are companies out there that are trying to build what they call AI scientists. And actually, I wrote a piece in The Conversation, if you're familiar with The Conversation, last year about one of these, and asked some of the ethical questions that you've asked me. So, you know, do we want a scientific ecosystem where we have machines that are generating hypotheses, running experiments, analyzing the data that comes out of the experiments, writing up the paper, peer reviewing their own papers? There's a machine from a company called Sakana AI that is doing this right now. And I don't think we want this, right? I think, you know, it has massive implications for the scientific ecosystem. It has massive implications for peer review. Who's going to peer review all of these papers that are generated in 15 minutes and $15, right? I won't be. In fact, I just reviewed a bunch that were written by ChatGPT last weekend, and was not very happy. So, you know, I think we really have to ask. But for me, the objective is not to replace the human. It's to help the human to be more comprehensive in their systematic reviewing, and to help them make it more efficient so that we can keep up with the massive increases in our scientific productivity more effectively.
Yeah, my best guess is like I could use AI to generate a hypothesis, then test it myself, like to
yeah, so that's exactly the idea behind the literature-based discovery, right? So if you hypothesize that fish oil might be connected to Raynaud's disease, it doesn't tell you it is. It's just a you know, a good guess based on the information that's in the literature, which in some ways is what your brain is going to be doing anyway, right? When we do a systematic review and we or a review of the literature, and we identify a gap, that gap is always a new question, right? It's a research question, and it doesn't answer the question, but because then you have to go through the process of working out, well, how do I answer this question? Do I need to do a clinical trial? Do I need to, do a molecular experiment? How can I test this question in order to gain evidence for the answer? But it doesn't actually tell you the answer. It just gives you the hypothesis.
Okay, thank you very much. Thanks.
Perfect. Is there any more questions online or in the room for Dr. Verspoor? Okay. Well, thank you very much, Dr. Verspoor, for such an interesting topic and sharing everything that you have learned about this so far. Right now, there will be a 10-minute break with snacks in the hallway for those of you who are in person, and you can come back into the Maple Leaf room around 3:30, both in person and online, for the keynote speaker session featuring Dr. Mamatha Bhatt, who will be speaking on optimizing equity and outcomes post-transplant using AI. So let's give just a round of applause again for Dr. Verspoor, and enjoy your break.
Thank you. Bye.
Bye-bye.